Good day to everyone. I'm Hazelle Jae Abanilla with my partner Luis Alfonso Dañez and our thesis advisor, Dr. Christopher Ray Pamintuan, will be presenting the results and findings of our study entitled Stacking of Novel 3D Printed Hexagonal-Prism Plant Microbial Fuel Cells Growing Water Hyacinth. A plant microbial fuel cell is a novel bioelectrochemical system that uses the root exudates of plants for microorganisms to metabolize, generating electricity as a result. Several studies showed that PMFCs could not generate sufficient electricity or have low power densities. An attempt to optimize the power performance of PMFCs involves stacking, or connecting cells in series, parallel, or combination of both. The study's objectives are to determine the power production capacity of PMLC assemblies growing water hyacinth that incorporate a 3D printed magnetic hexagonal prism design composed of alternating anodes and cathodes, to examine the effect of varying patterns and connections on the performance of PMFCs, and to identify the optimal stacking configuration and connection of the PMFCs based on the maximum power and power density produced.
In this study, a novel design of 3D electrodes was done using Fusion 360. The structure is analogous to that of a honeycomb, with 6 sides of the hexagonal prism cell connected to other cells. Firm contact between cells can be established using magnet pockets incorporated into the legs of the electrodes. The PMFC is composed mainly of 3 components: the anode, separator, and the cathode. The site of the oxidation reaction is the anode, whereas the reduction reaction is the cathode. A separator component is placed between the electrodes to isolate both reactions. All members of the PMFC were 3D printed. Both electrodes were printed using polylactic acid conductive filament, while the separator used a non-conductive PLA filament. Shown also in another figure is the disassembled version of the PMFC. Cube-shaped neodymium magnets were used inside the pockets of the legs of the PMFCs. Each of the legs of the PMFCs were long enough to fit 5 pieces of neodymium magnets. Furthermore, the magnet's polarity was also considered so there would be no repelling force between the PMFCs. As demonstrated by their respective polarities, the researchers have thought of a way to connect 2 adjacent sets of magnets so that attraction will occur when placed in their respective PMFC legs. Following this pattern shown in the figure is how the neodymium magnets were arranged for all the PMFCs. The arrows represent the direction of attraction of the neodymium magnets. Medium-sized water hyacinth plants were bought from a hydroponic farm and were individually placed in each of the 15 cells. The substrate used in the experiment was sourced from a fishery pond. Before the experimentation proper, an acclimation period of 7 days was implemented to give plants enough time to adapt to the sudden changes in their environment. Each system is placed inside a 50-liter plastic container long enough to account for the space taken up by all the cells. Water gathered from the fisher pond is filled right below the legs of the PMFCs and inside individual cells. Shown here is a sample connection for recording readings in an individual and a stacked PMFC setup, along with how the clips are connected to the PMFC for a stable and correct reading. With the design, there are several ways the positive and negative terminals connected to the multimeter can be attached to the PMFC. To determine the optimal placement of the clips, a test was performed to see if there were any differences in the readings of the cells at varying locations. Only an individual cell was used for the first part of the test. The legs of the cathode were numbered from 1 to 3, whereas the legs of the anode were given the legends A to C. The second part of the test involved 2 stack cells with cathodes 4 to 6 and anodes D to F for the other PMFC. Tables 1.1 and 1.2 show some possible combinations for each triad. Each system contained a plant and was individually observed for 5 days. Flags were placed on distinguishing each cell. Cumulative stacking was employed, wherein the PMFCs were continuously measured while increasing the number of stacked cells by 1 for every triad. The series connection was made by connecting the opposite electrodes of each PMFC with each other. In contrast, parallel connection involved connecting the same electrode of each PMFC with each other. Cells were stacked in series and parallel at different patterns, namely linear, branched, and closed. Furthermore, the behavior of PMFCs during one-cell increments for the experiment was quantified to assess the optimal number of stacked cells that can produce effective readings. Figure 6.2 shows the patterns for the series and parallel configurations. The third part of the experiment involved illustrating the behavior of all cells stacked in one cluster when series and parallel connections are combined. As for this test, it is focused on how power generation can further be amplified by changing only the connections of a single pattern. Figure below shows the variations in terms of the connections of the clump cells, where the red sides depict the anode and the blue sides for the cathode. Shown in the figure are the patterns employed during the experimental proper. To distinguish the connections when cells are oriented to change configuration, connections between cells are highlighted as red when in series and blue when parallel. A waiting time of 10 minutes was designated to allow systems to reach stable values. A polarization test was performed on the three of the best performing individual cells, all series and parallel connection configurations, and the best performing clumped configuration. Voltage and current readings were measured at varying external resistor values ranging from 220 ohms to 3.9 mega ohms.
For the first part of the study, we can see that there is an uneven power generation when cells are measured at different combinations of anodes and cathodes. This can be attributed to several factors such as the retarded diffusion of electrons, differences in internal resistances, and many more. Another notable explanation that was observed in a similar study was that there is an uneven biofilm distribution. This resulted in lower electron transfer efficiency and power generated. Figure 8 compares the voltage, current, power, and power density values of the 15 PMFCs. And although they are all growing the same plant and were subjected under the same environmental conditions, differences in their performances can be observed. This can be caused by the differences in their plant characteristics, the microbial activity, substrate concentration, and anode and cathode kinetics. This part of the presentation deals with the results of when cells are cumulatively stacked in series. As shown in Figure 9, the actual voltages for all three patterns were much lower than expected when stacked cumulatively. These voltage losses are observed in cells stacked in series as a result of charge or voltage reversal and high internal resistance from slow electrokinetics. On the other hand, measured current values were significantly higher compared to the theoretical values. High current values can be attributed to the system's high surface area to volume ratio due to the use of miniaturized PMFCs. The average individual voltages were much lower when compared to that of its theoretical voltage. However, the average individual and theoretical current values were equal at 2.82 microamperes. In general, there are no significant disparities between the patterns. These results showcase the reading's stability after repeated voltage and current measurements for all patterns. As for stacking cells in parallel, higher voltage values were observed for all three parallel stack patterns, the highest of which was generated by a parallel linear pattern at around 163.92 millivolts. Current values, however, did not show any increase in trend and were lower than the theoretical values. The highest average current for parallel was generated by a branched pattern which is equal to 12.1 microamperes. From the results, we can see that parallel stacking helps prevent voltage losses but cannot generate high current values. This is because of the resistance of electrons to flow through electrodes and mass transfer limitations. Individual cells performed slightly better than expected in terms of voltage, but current deviated greatly from the theoretical value. Reconfiguring parallel patterns can have a slight noticeable effect on the stack voltage and current on the PMFC system. In terms of the power and power density values generated when cumulatively stacking cells in both series and parallel, the highest power and power density was generated by a series branched pattern. A consistent behavior for both power and power density values can be observed in all series and parallel configurations. An increase in power generation arises from the increase in the total anodic surface area available due to stacking. However, power density decreases as a result of substrate concentration losses due to the limited mass transfer between the electrodes and the biofilm. The decrease in the power density can also be associated with the idea that there was no considerable improvement in power as the surface area increases. The power and power density values of all stacked patterns were much higher than that of an individual cell. The highest power and power density values were generated by a series branched pattern. Parallel stacks are associated with internal losses due to different current values of individual cells. In this regard, the series branched pattern is revealed to be the most efficient in terms of power and power density generation. It is proven by the results, as shown in Figure 14, that stacking cells, may it be in series or parallel, improves the overall power generation. When cells are clustered, its connections are a combination of both series and parallel. As shown in Figure 15, combination 1 is the best performing hybrid configuration, as it generated the highest average voltage, current, power, and power density values. Stacking cells in a cluster with series parallel connections improves the voltage, current, and power of the overall system, but generates a very low power density compared to that of an individual cell. The power density values of clustered stacked cells are nowhere near those of pure connections, using the series branched and parallel closed patterns as a basis. We can also see that in Figure 15, switching the connections from combination 1 to combination 2, 3, and 4 caused a decrease in the voltage, current, and power density values. Changing connections have a similar effect to when there is a change in external resistance, and can therefore negatively affect the microbes in the anode. As a result, decrease in performance can be observed. Reorienting cells from series to parallel or from combination 1 to combination 2 causes a deterioration in the performance due to the effect of this change on the diversity of the microbial communities within the anode. Electrochemically active bacteria required a recovery period to adapt to the changes in connection, which is why there was no increase even though cells were reoriented back to series. The last part is the polarization of all the systems dealt with in this study. We can see that in Figure 17, a common behavior or trend can be observed for both voltage and power density with increasing current density. The voltage drop could be imposed by polarization losses, whereas for power densities, they peak at a specific external resistor value, signifying the system's maximum internal resistance according to the Jacobi theorem. Beyond this point, higher external loads resulted in a stepwise decrease in the power exerted. High external loads restrict microbial activity, resulting in difficulty in the transfer of electrons. Fuel consumption also decreased after the optimal external resistor value was reached due to the shift of microbes to a less electrogenic culture. Table 3 summarizes the polarization test results of the study, from which we can deduce that a series closed configuration generated the highest maximum power density, since series stacks are often associated with lower internal resistances and therefore higher power densities are produced. On the other hand, a clump pattern has the highest total internal resistance as a result of more unwanted losses from combining connections in one stack. Therefore, it produced a lower maximum power density compared to that of an individual cell.
The study determined the efficiency of stacking 3D-printed hexagonal prism PMFC regarding power generation. Incorporating this design proved difficult as there was no uniform voltage and current readings distribution across all sides of a cell or cell stack. It was found that stacking cells in series and parallel cannot replicate the behavior of electronic circuits due to their biological nature. The maximum power densities were found for a series branch connection and as well as for a closed parallel connection, which was relatively lower than the average value of the individual cells. Combining series and parallel connections in a single cluster caused the power density to drop compared to an individual cell. Changing the connections of the honeycomb stack resulted in a deterioration in the system's performance. Polarization tests revealed that the clump pattern had the highest internal resistance. At an optimal external load of 68 kiloohms, the closed series pattern came first with the highest maximum power density. This study is the first to utilize a multi-anode cathode hexagonal prism PMFC design and conduct stacking efficiency studies on a clustered pattern. Improving the efficiency of PMFCs through stacking should therefore be sought after due to the difference in the results from other studies.